In this project for AP Bio, I will talk about proteins. Proteins are large biomolecules or biomacromolecules that are essential for organisms to live and to function. They are proteins that catalyze metabolic reactions, perform DNA replication, respond to certain stimuli, transport molecules, and provide the build and structure for the cells of the organisms. Many proteins are enzymes that catalyze biochemical reactions, and other proteins play a significant role in cell signaling, the immune system, and cell adhesion, which is the contact and the attachment between two cells. So proteins don't have to work alone. They can work together on a specific activity, and they can also associate with one another to form large protein complexes. Proteins can also interact with many other different types of molecules, such as lipids, carbohydrates, and DNA. So to understand just how small and abundant these proteins are in organisms and in the human body, one human cell has about 1 to 3 billion proteins in it. And to put that into perspective, the human body has an estimated 30 trillion cells in total. That's a lot of protein, and there's more, because the human body contains about 100 trillion bacteria cells, which is more than human cells, and an average bacteria cell has about 22 million proteins in it. So what exactly are these proteins? Well, proteins are large chains of amino acids bonded together by peptide bonds, and the differences between proteins is primarily determined by the sequence of these amino acids. They come from our genes, from our DNA, because the sequences of nucleotides in our genes determine the sequences of the amino acids of our proteins. So there are about 20 different standard amino acids, although there are some organisms that have more than that, and the average protein contains about 438 amino acids. But of course, this is an average and there are extremes. The largest proteins known to scientists today are titans. Titans are found in the muscle, and they contain up to 27,000 amino acids. And written out, the full name of titan is also the longest word known out of any language. The proteinogenic amino acids have an alpha carbon, or a central carbon, to which a carboxyl group, an amino group, and an R group, or side chain, is added. The exception to this is proline, because near the nitrogen end of proline, a ring is formed that forces the carbon-oxygen part and the nitrogen-hydrogen part into a fixed form. So the R groups determine the differences between the amino acids, and they come in a lot of variety. And ultimately, the combined effect of the side chains in a protein determine the unique 3D structure that it will have. So to make the chains, amino acids are connected through covalent bonds, and specifically peptide bonds. And then a peptide bond, the carboxyl end of one amino acid, bonds to the amino end of another amino acid. The carboxyl end loses an OH, or hydroxide, and the amino end loses an H, or hydrogen. And the OH and the H form H2O, so water, as a byproduct, and that's why the peptide bond formation is a dehydration synthesis. So the chain of amino acids is called a polypeptide. Shorter chains of amino acid, that contain about 20 to 30 of them, are just called peptides. The amino acids and other components and other properties, they usually cause folding into this very precise 3D structure. The shape into which a protein naturally folds is called the native conformation. And because of the many properties of the amino acids, most proteins kind of fall into this native conformation without any assistance. But there are some other proteins that need extra assistance from molecules called chaperone molecules. So the different structures that proteins can have based on many different factors are classified by biochemists into 4 different groups: the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. The primary structure is the most straightforward. It is the amino acid sequence. The secondary structure is made up of the repeating local structures in a polypeptide chain that form because of hydrogen bonding between distant amino acids. So these interactions of hydrogen bonding usually result in a helix structure, an alpha helix, or in sheets of strands called beta sheets. And because secondary structures are local, different secondary structures can exist within the same protein. So the tertiary structure, also called the fold of a protein, determines the basic shape and overall function of the protein. It is formed by the relationships between secondary structures, by the different properties of the R groups and the side chains, and it is stabilized by non-local interactions. So these interactions include a hydrophobic core, salt bridges, which are electrostatic interactions between charged parts of the polypeptide chain and of these side chains, disulfide bonds, which are the bonds between sulfur atoms, and hydrogen bonds. For example, some R groups could be hydrophobic, which means that they don't like water because of their nonpolar structure, and some R groups might be hydrophilic, which means that they like water because of their polar structure. The quaternary structure consists of several different polypeptides, called protein subunits, and you can think of it as a single protein complex. So proteins are not rigid structures with a fixed shape. The atoms in the protein can move a little bit, and the overall structures of the proteins can change as well. So the key characteristic of proteins that allows them to have all of these different functions and so much diversity in their functions is their ability to bind other molecules extremely tightly and extremely specifically. The binding site of a protein, which is the area that binds to other molecules, is determined by the side chains of the amino acids in the protein and therefore the tertiary structure. So what do proteins bind to? Well, other molecules of course, but also to other proteins. And when a protein binds to an identical copy of itself, the same protein, they can oligomerize. They can form an oligomer, which is a large molecule that consists of multiple copies of its individual units that are called monomers, and the result of this formation are fibrils. So proteins interacting with other proteins can regulate enzymatic activity, control the progression through the cell cycle leading up to mitosis, and also allows for the formation of large protein complexes by the assembly of proteins, and the ability of proteins to induce conformational changes in one another allows the cell to develop these immensely complicated signaling networks. Alright, let's now talk about the functions of the protein and what they all do. Enzymes. Enzymes are proteins that catalyze biochemical reactions. They perform many different functions, for example with DNA manipulation like transcription, repair, and replication, such as for instance helicase that opens up the DNA strands for transcription and spins about as fast as a jet engine. And overall, 4,000 biochemical reactions are known to scientists today to be catalyzed by enzymes. Many reactions also simply would just not happen at all without enzymes. Cell signaling. In cell signaling, many proteins are involved. Proteins such as insulin are called extracellular because they take a signal from the cell they were originally made in, synthesized in, and then they take it to another cell in distant tissues. Antibodies. So your immune system has a bunch of antibodies, and these are proteins that bind to antigens or foreign substances and destroy them. Ligand transport proteins. To start, what even is a ligand? A ligand is any substance that binds to another chemical to form a larger chemical and a larger complex. Ligand transport proteins, they bind to small molecules and then transport them to other locations in a multicellular organism. And an example of this is hemoglobin. Hemoglobin is found in red blood cells. It's responsible for giving blood its red color, and it transports oxygen from the lungs to other tissues and organs in an organism. It occurs in all vertebrates, and one red blood cell has about 70 million of these hemoglobin proteins. Transmembrane proteins. Transmembrane proteins are also examples of ligand transport proteins, and they allow certain molecules or ions to enter the cell via the cell membrane. Structural proteins. These proteins are there to give rigidity and structure to biological parts of our cells and our body. Motor proteins. Motor proteins are the molecular motors and machines of the cell that move within the cytoplasm. They take chemical energy in the form of ATP and convert it into mechanical energy, and they can also have structural functions. Motor proteins are imperative for many reasons, but especially because they allow the motility or movement of single-celled organisms and sperm cells of multicellular organisms. So motor proteins also have significant roles in intracellular transport and muscle contraction, and examples include myosin, kinesin, and dynein. Kinesin is especially interesting because it actually has two legs and walks along these microtubules that look like highways and serve as highways. Animations of a walking kinesin are just absolutely mind-blowing. As a last note for this section of the video, proteins do not exist forever. For some, the lifespan is minutes, and for others it's years, so the lifespans vary, but the average lifespan of a protein in mammalian cells is 1 to 2 days. Alright, so to finish up the video, let's talk about eating proteins, nutrition. So now that you know how important these proteins are, of course it should be obvious that we need these proteins. So whereas most microorganisms and plants can synthesize all 20 amino acids necessary to make all the proteins, animals cannot synthesize all of the 20 amino acids. So the amino acids that we cannot synthesize on our own are called essential amino acids, and in order to get these essential amino acids, we need to consume protein-containing foods. When we consume protein, the process of digestion breaks these proteins down into the individual amino acids. So once we have our essential amino acids, three things can happen to them. They can be used for protein biosynthesis to create proteins, they can be converted into glucose in a process known as gluconeogenesis, or they can be used for the citric acid cycle, or the Krebs cycle in cellular respiration. And that wraps up this section and the video. Thank you for your attention.